I'm ready now to talk about some additional examples of parasite-induced alterations in host behavior. And these three examples actually pertain to parasites that do not infect humans and are not really important in that regard. However, I think you'll agree that they're all very interesting from a biological point of view. So first, an introduction to the acanthocephalans. The acanthocephalans are helminths, and they are known commonly as thorny-headed worms because, as you can see, this proboscis is actually covered with little thorn-like projections, and you can see them over here as well, that allow this parasite to anchor into the mucosa of the definitive host. And these parasites are highly specialized for a parasitic lifestyle, and basically are bags of reproductive organs that are used for producing eggs and progeny. There are no, as I said, important acanthocephalan species that cause disease in humans. However, acanthocephalans do infect a variety of mammals and birds and reptiles, so they're very common in nature. And a very typical life cycle of an acanthocephalan involves an insect intermediate host and a vertebrate definitive host. And it's a fairly simple life cycle. So just to give you one example of an intermediate host that is used in aquatic life cycles of acanthocephalans, this organism here is an amphipod, commonly referred to as a side swimmer, and these are found in freshwater habitats. And these amphipods become infected with acanthocephalan larvae through ingestion, and then these larvae develop inside the host. Now, for this life cycle to continue, this intermediate host containing the larval acanthocephalan has to be ingested by the definitive host. And so what you see with these acanthocephalan species is that often the insect host undergoes both a color change and a behavioral change. So for instance, if these species are very dark in coloration, sometimes, here you can see one that's very brown, sometimes they're completely black, and once they're parasitized, they lose pigment and are sometimes completely white and much more visible to their predators. In addition to the color change, which makes them more visible, the presence of the parasite seems to also alter their swimming behavior, again facilitating not just identification but availability to the definitive host. Next, I have two examples of parasites that are known as digenetic trematodes. And the term digenetic refers to the fact that these trematodes, or flukes, require at least two hosts. Some of the life cycles, for instance this one, involves more than two hosts. So in this life cycle, and the parasite's name is Dicrocilium dendriticum, there is a first intermediate host, which is a land snail, a second intermediate host, which is an ant, and the definitive host, which would be sheep or cattle or other grazing mammals. The life cycle works like this. The grazing mammal is a definitive host, and these flukes reach maturity, and the female flukes begin to produce eggs, and the eggs are released into the environment. And this is a terrestrial life cycle. The vast majority of digenetic trematodes actually use an aquatic life cycle, and very often an aquatic snail species as its first intermediate host. But here we have a parasite that is adapted to this terrestrial cycle. So we have an egg that is released, the larval stage is inside the egg, and the snail consumes this egg as it is grazing and picking up organic material. There is a fairly complex developmental progression of the parasite inside the snail, culminating in the production of the next larval stage. And in aquatic systems, this larvae is a swimming stage. It even has this tail that allows it to swim in water. But this parasite has adapted to this terrestrial life cycle. And what happens is the snail actually releases these larvae in these slime balls. So the slime helps to keep the larvae hydrated and protected from desiccation, at least for some period of time. And along comes the second intermediate host, which is the snail. The snail is attracted to the slime trails of snails, and as they're ingesting and picking up material, they're also picking up these slime balls full of larval Dicrocilium dendriticum. What happens next is that the larvae then undergo additional developmental progression, and this stage enters the cerebral ganglion of the ant, and the presence of the larval trematode in the cerebral ganglion alters the behavior of the ant, and the ant tends to climb up on vegetation, up to the tips of vegetation, and most surprisingly, the presence of the parasite actually triggers the ant to close its mandibles on the plant, and they're completely unable to release their mandibles. So overnight, the ants stay up on the vegetation, and the next morning, the grazing mammals come along and feed, and in the process, pick up these ants. On week 1, I posted 3 YouTube videos, and 2 of those videos depict in a very short, sort of entertaining way these behavioral alterations, and there is one on Dicrocilium dendriticum. In addition, the second video that I have posted is on Leucochloridium paradoxum. So if you've already watched that video, you know the punchline, and that is that Leucochloridium paradoxum in the snail host has a very, very striking effect, one that clearly promotes predation on these infected snails. So again, it's a digenetic trematode, and it has a land snail as an intermediate host, and birds serve as a definitive host. And when the snails become infected with this parasite, the larvae migrate up into the antenna. So if you are familiar with snails, you know that this is not the normal appearance of snail antennae. And not only do these larvae change the appearance of the antennae, so they're swollen and they're pigmented, they have this sort of striped appearance, and they look very, very much like a caterpillar stage for an insect that would be quite a tasty treat for a bird. So not only do you have this pigment and this bright coloration, but you also have a pulsating activity, and birds can readily see these. And as you could see from the video, sometimes they come and just snap off one of the antennae. Sometimes they come and ingest the entire snail, and it's a very effective way of passing on this infection. So this concludes our overview of the topic of parasite ecology, and a specific emphasis here on the alteration of host behavior by parasites to facilitate transmission. And at this point, there are review articles posted on the Blackboard website that everyone can download and read. And once I give folks a chance to go over these lectures and read those papers, we'll have some time to discuss those articles in our blog format. So